like Young Scientist Award, a Fulbright Nehru Postdoctoral Fellowship, a Young Women Scientist Award, and she is a fellow of Plante, and recently she has been selected for INIAS Fellow of Indian National Science Academy. She has published more than 40 publications in peer-reviewed journal, and she is expert in the starch kinetics area, and she has developed several matrices linked with this. So I think in brief, so this is introduction of Veda, and today she is going to give a talk on assessing the starch kinetics of pearl millet flour. So I think with this brief note, now I invite Dr. Veda Krishnan to please continue with the invitee talk.
Thank you, sir.
Veda.
I'm audible?
Your voice is not clear, Veda.
What about now, sir?
Yeah, it's okay.
A very good morning. So thank you for that overwhelming introduction, and a very good morning to each one of you. And even though we had a very small introduction in the inaugural session, so can we have a very brief introduction of the participants also, so that I can like audit myself that how I have to have my talk.
Please.
So if that's okay, just 2 lines about like, I can have some familiar faces here, but still name, affiliation, and that what is your research domain. Yeah, Mohan, you can start.
Good morning. I'm Yatiraz Kare from CNQV Jabalpur Krishi Vigyan Kendra. My research will be agriculture extension.
Okay, sir. Okay.
Good morning, ma'am. Myself, Dr. Aradhana Kumari, posted in College of Agriculture, Jawaharlal Nehru Krishi Vishwavidyalaya. And hello.
Yes, ma'am, you're audible.
And my specialization is plant physiology, and I'm working in the area of drought stress and related work.
Hello. Hello.
Yeah.
Yes, Dr. Sanjay.
I'm Dr. Sanjay Jain from Krishi Vigyan Kendra, Baitul, under Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur. I am basically master's and doctorate in plant breeding and genetics.
Okay.
Good morning, madam.
Yes, sir, please.
Good morning, madam. Sir, myself Raghavendra, and I am in Achal Engineering Agriculture University, Andhra Pradesh, along Guntur, madam. I am in teaching, madam, not in research. And I had met you in 2018.
I remember your face. Yes.
It's a 21 days training program.
Yes, sir. Yes. Okay. Welcome back.
Good morning, ma'am. I'm Mohan Chauhan. I'm from University of Agriculture Sciences, Bangalore, working as assistant professor of biochemistry in the division of plant biotechnology.
Okay.
Good morning, ma'am.
Yes, please.
Good morning, ma'am. I am Dr. Anita. I am working in the Department of Nutrition Biology, Central University of Haryana, and my expertise is nutritional composition of foods and food formulations from underused crops of Haryana.
Good morning, ma'am.
Yes, ma'am, please.
Myself, Dr. Geeta Singh, working as scientist in Krishi Vigyan Kendra under JNQ, Jawaharlal Nehru Krishi Vishwavidyalaya. My specialization in agriculture extension. Okay, thank you, ma'am.
Okay.
Good morning, ma'am. My name is Mahesh Kumar, posted at Kazi Jodhpur, plant biochemistry.
Okay. What are you working on now?
I am working on abiotic stress management on arid crops and nutritional quality improvement.
Okay. Yeah. Please jump in, please. Next.
Good morning, ma'am. Myself, Dr. Alpna Sharma from JNKVV Krishi Vigyan Kendra Schedule. And basically, I'm a food scientist and posted as a home scientist in KVK and working for the nutritional benefit of the tribal community.
Okay, wonderful. Okay.
Thank you.
I think you should be a little bit fast.
Yes, please.
Yeah.
Akansha. Yes, Neeru ma'am, please.
Hello, good morning, ma'am. Myself, Dr. Akansha Tiwari, and I'm working as assistant professor at College of Agriculture, Azamgarh, Acharya Narendra University of Agriculture and Technology.
Okay. Yes. Dr. Neelu.
Good morning, ma'am. Myself is Dr. Neelu Vishwakarma. I am working from Krishi Vigyan Kendra, Jabalpur. My interested area is value addition and nutritional security. Thank you so much.
Welcome. I think few more are there. I think anybody want to introduce or should we start?
Hello, good morning, sir. Good morning, ma'am. I'm Dr. Praveen Zadav, working as an assistant professor at Dr. Punjab Rao Deshmukh Agriculture University, Maharashtra. Presently, we are working on some quality as well as biotic stress related traits in soybean.
Okay.
Thank you.
Okay.
Good morning, madam.
Yes.
Myself, Dr. Ellen Yogesh. I am from University of Agriculture Sciences, Raichur, Karnataka. I'm working on millets, madam, millet crop improvement, developing millet varieties.
Okay.
Thank you.
Anybody else, or can we start for the day? Ranjan sir, can we start then?
Yeah, please, ma'am. Please go ahead.
Okay. So a very good morning. I tweaked the title a bit because assessing the starch kinetics, we will be doing definitely in the practical session. Can I first request everybody, can you please mute yourself? So that will be easy for us. Yeah. Please mute yourself. Thank you so much. So today I am going to discuss a bit more because I got to know some are breeders, some are extension people, some are working in real nutrition, some are working on other domains. So today's talk, I think that it can touch many of you because we are addressing a very important nutrient, that is starch. So starch, I myself consider as carboholic because I have a craving for starch, not only in research. So the starch, especially, we know it's good for nutrition and many more. It has a lot of economic importance. And other than that, as an extension scientist, when you go and meet people, we know the starch is one commodity mostly considered also as a culprit for many diseases. Nowadays, we know the diabetic problem and its associated complications. So normally we say it is from the high glycemic diet. So it is connecting to our life in many ways. So that's why we wanted to know that how pearl millet is good in this case. And I think you all are undoubtedly agree that pearl millet has a nutritional supremacy, right? Pearl millet is rich in its nutrition in terms of its components when we say the nutrient density score. And since decades, like as we know, it's an archaeological staple. Since decade, it is known it is diabetic friendly. But see, what we say, systematic studies has not done in this crop till date. So under this niche area of excellence program, even though the program was more addressed on its processing attributes to improve the shelf life of pearl millet, but our honorable director and our head, Dr. Shelly Praveen, they also want to address that what is the nutritional spectrum of pearl millet. Dr. Dhananjay, can you please mute yourself? Sir? Okay, let's move on. So that's why, as you all know, because I'm not getting into the detail that how pearl millet is rich or superior in this case, because I think already in the first lecture, Dr. Shelly Praveen has already stumped you all with the details of its nutritional spectrum. But what we want to know more in detail that till date, the research has been carried out in extracting the nutrient and then analyzing it. Okay, but that is not the real case when we try to translate it into an in vivo condition. In an in vivo condition, we are not eating only starch. We are not eating only protein. We are not eating only the minerals, but we are eating the food as a matrix. So that's why it's very important to understand that the nutritional studies have to go one step ahead by addressing the food matrix. Because in matrix, all the nutrients are together and the interaction, and there are a lot of things that is going on inside which we cannot translate in our basic nutritional studies. So that's why we wanted to go one step ahead in understanding the matrix and how the matrix is causing a nutrient bioaccessible and ultimately how it is going to be bioavailable. So that we cannot study when we isolate the nutrient independently. Okay, so till date, most of the studies is like we say that this much starch content is there, this much protein content is there, but that is all after extracting that one particular nutrient, which is not an ideal scenario. So that's why, as we say, this is the evolution of nutritional studies. And when we did a literature survey, what we saw that till most of the research, as you can see, 98.5% of the research in pearl millet is only the tip of the iceberg. It is only understanding the composition, the spectrum, and also characterization. Some of the characterization like digestibility of the protein or digestibility of starch, or some studies on high iron, high zinc varieties. So most of the studies are limited till this much. But the real attribute or the ultimate indicators of nutrition is the food matrix, which is not at all studied in case of pearl millet. The dynamics that is happening among the nutrients, how the presence of starch and protein is going to affect its digestibility, how the minerals are going to alter it. So it's not been addressed. The third is bioaccessibility of your nutrient. Bioaccessibility means how your nutrient is released from the matrix, how easily it is released. So that is very important. And finally, how long it will take to reach into your blood system, that we normally say as bioavailability. So all these things we have to address, and in that way we started our study. So let me brief about food metrics, what we mean. So food metrics is the spatial or the physical domain embedded in the component. Directly or indirectly, it can impart a function. So as we know, if it is a rice, 90% is the starch, and very few percent of protein and lipid is present in rice. But in case of pearl millet, it's a bit more complex, because there, along with the starch, you have protein also, and you have more phenolics, and you have about 5% lipid also. So we cannot expect the digestibility of starch in rice will be similar to pearl millet. But if you extract the starch from both sources, and when you do a digestibility assay, it will be same, because at the end of the day, biochemically, starch is a composition of amylose and amylopectin, which will degrade to give you glucose. So this is not the right way to do a starch digestibility kinetics, which we are going to discuss today. So you have to consider the food matrix itself. You have to take the tissue itself, like it is a roti, if you have to take the roti itself, or it's a powder, you have to take the powder itself, or a cooked rice itself. Okay, rather than extracting the starch and doing the digestibility. So that's why food matrix is very important, because the metrics vary in different scales, from nano to the ultimate scale, if you see, the matrix is different. And all the nutrients, what you say, in molecular level, if you go, again, these are all semi-crystalline polymeric structures. So their orientation, their disaggregation, their deformation. So as a biochemist, I am more interested in all these things. So it's entirely different. Okay, so till date, we have not seen our food in this angle, which is also very much important. And why it is more important? I started my journey in 2011 as a scientist, and 2012 when I joined the group of Dr. Archana Sachdev. And under ma'am, in that group, we were working on phytic acid. So we know phytic acid is an absorption in which, okay, maybe you might be getting like 20 ppm iron, but in the real, if you're eating the soybean as a food, because of the stick acid, definitely that much mineral release is not happening in our body. Okay, so that is the need of understanding, because in whole food there are different type of interaction that is happening. Sometimes it could be complementation, it could be a synergy, it is a positive effect, it could happen. For example, we did in red rice. In red rice, we found it is very good for anti-diabetic because it has a particular phenolic and the starch quality. So there the matrix is synergizing. Sometimes it is an enhancement effect, but sometimes it can have a masking effect or an inhibition effect. So all these things you can only study if you are studying the food matrix. So we can't understand starch alone. So that is my first point that I want to put forward to you. So we have to study the matrix. With the matrix only we can understand that is what is happening. So as it's a very heterogeneous group, our earliest memories about starch, the earliest memories about starch, I think it's this experiment that we all studied in the class in our, I think high school level, where we studied the photosynthesis which produced starch. And with the help of iodine, we detect the presence of starch. And we all know that starch, it is one of the most major macronutrient, and it is composed of glucose units, and it's a source of dietary energy. More than 60 to 70% of our per capita dietary energy we are getting from starch. Iodine test we all have done in our classes, where it converts into blue and we say the presence of starch. And along with that, we know starch is a polymeric carbohydrate, and it has two components: amylose, which is linear, glucose with alpha-1,4 glycosidic bond, and amylopectin, which also has an additional alpha-1,6. So amylopectin is a branch structure and amylose is a linear structure, which together form this polymeric compound called starch. So that is the basic understanding that we all have. And more than that, what is the importance? Because, like when I have to market the molecule of starch, I want to make you realize that what is the importance of starch. So the importance of starch is that we are consuming it in a daily basis, not only from the staple cereals, but also from other non-conventional sources, which we normally we don't aware about it. We never think that we are eating mango and we are getting some kind of a starch. Definitely sugars are there. But so we have a lot of conventional sources of starch, non-conventional sources of starch, without which we cannot live, because that is the main dietary source of our energy. And along with that, on the industrial the starch is also has a lot of economic value, because starch is well used in food industries. It has a thickening agent, gelling agent. It is nowadays used in many of the fortified products. So the revenue that we come across from the starch as a commodity is also very important. And other than that, evolutionary also, starch has a lot of importance. When you can go through this paper in which they mentioned that the evolution of human that happened in the Pleistocene age is mainly because of their consumption of high energy cereals or staple cereals. Because for the development of brain, you need more metabolic energy. And for more metabolic energy, you need energy rich food. So starch is involved even in our evolution, and that's why it is one of the most important molecule. It is not only just giving energy to you. And let me here just briefly explain the complexity of the molecule starch. because we don't normally get into this detail, but when we have to understand the digestibility of the starch, you also have to understand that what is the hierarchy of starch that we normally see. So as you can see in this figure, in a grain, for example, a rice grain or a pearl millet grain, we normally see starch as granules. But in that, when we go in depth, in detail, we can see these each granule are composed of growth rings. And these growth rings, as you can see in this figure D, is composed of blockade structure. So what are these blockades? These blockade structures are composed of two types of, we call them lamellae. Okay, one is a crystalline lamellae and another is an amorphous lamellae. Crystalline means it's very compact. Okay, and amorphous means it is loose. So the crystalline part is composed of your amylopectin double helices. So amylopectin is branched and it is combined to form the crystalline, very compact part of starch, and amorphous is very loose part of the starch. So whenever digestion happens in our body, the digestion starts from the amorphous region. That is happening in case of starch. And then if you get into the detail, you can see that amylopectin forms superhelices, and amylopectins are mainly of two types, L type and S type, based on the branching diversity, as you can see here in the I sub image. L type amylopectin, S type amylopectin. So all our cereal types that we see, they have different type of branching, and based on that, its digestibility varies. So that's one point that I want to put forward here. Same way, amylose also. So amylose, we know it's linear, but it is not existing in a linear, in a molecular form. So it is existing in a crystal form. So in the crystal form, along with the water molecules, it exists different crystal structures or lattice structure. So it can be an orthorhombic A-type structure, or it can be a hexagonal structure, so which we can analyze by XRD method. So based on that, the digestibility also varies. So my point here is, starch is not as simple as we see. It is very complex, and the complexity at this molecular level determines its digestibility. And it's just, you can see that how it forms an A-type crystallinity, which is mostly common in your cereals. The rice, even in pearl millet, we have seen it is A-type. But in case of B-type, we normally see in tubers. In potato and other tubers, it is B-type, which is less digestible. Then sometimes there is another type that is called C-type, which is a combination of A and B. In case of bean, kidney bean, the type of starch that is present in beans are C-type crystallinity. And the last type, which is very recently called identified, is V-type, which is digestion resistant starch. We have a project here with the DST SERB, in which we are understanding which are the rice varieties which are rich in high resistant starch. So resistant starch, if it is there, it has a different crystallinity because it is not digestible and it is not giving any glucose to your body. So now when we consume our starch, it could be from pearl millet, it could be from any source, what is happening in our body? So we know it is getting digested. So digestion happens in different steps. First, the polymer is converting into an oligomeric form like maltose or limit dextrins. Then amyloglucosidase enzyme will convert it into final glucose. So this is happening in our body in a chamber-wise digestion. So when we eat, the chewing or the mastication, there the amylase or the ptyalin, the salivary amylase, is mixed, and their first digestion starts. So that's why we know when we chew, we get a slight feeling of sweetness, which starts in our mouth. And then the complete digestion happens in your small intestine. And after the small intestine, the glucose gets bioavailable into the blood, and through the blood, it reaches your tissue. So we all have done our blood test. So we take some pure glucose source, and then after 2 hours, they ask you to get the test. So why? Because by that time, that pure glucose can get bioavailable and it can reach into your blood, and that blood can be tested to see that how much it is available. But if you are eating a food source, then it is a bit more complex because it's a matrix. So first your food have to be chewed, then it has to be ruptured for the starch to come out, then the starch have to be completely bioaccessible for the enzyme to attack, and then the enzyme have to degrade the starch into glucose. So an average, the time that is taken is around 4 hours to complete your digestion. It is not only the starch digestion because it is happening in toto, but it will take around 240 minutes, if you can say. So that's why when we do the starch digestible kinetics also, we give an in vitro timing of around 240 minutes. Okay, so that in 240 minutes, we can just try to see that similar digestion kinetics can we also do in an in vitro condition. So this is bioaccessibility and this is bioavailability. So if anybody has a doubt between these terms, let me tell you, bioaccessibility means the nutrient is released from the matrix. Okay, it all depends on the complexity of the matrix. If it is only starch rich, just like white rice, it is very easy to release. So that's why we say diabetic people don't eat much of white rice. But if it is a brown rice, definitely again the fiber content and the phenolic content is more. So the release bioaccessibility is less. It is in a very slow way. So that's why it is a better for diabetic people. Millets, definitely it is better because again, the food matrix is more complex. So that's why it is one hint that pearl millet or these coarse cereals are better for diabetic people or a pre-diabetic population, which is very rich in our country these days. And finally, the moment these glucose is released into your bloodstream, then it is bioavailable to your body and all your glucose sensitive tissues. We know that we have a lot of tissues like your muscles, your liver, where it is converted into the different forms. So all these tissues can only get the glucose once it is bioavailable. So this graph, I want to tell you that as you can see, the dotted curve is the pure glucose and the solid graph depicts the food source. So when we eat, this is a plasma glucose, so it's a human glucose test result. And if it is comparing a food source with a control glucose. So these kind of studies, we call it as glycemic index analysis. So after eating our food, we are analyzing the glucose and we are comparing it with the standard glucose content. So it's a index, it's a ratio. So that's why it is called glycemic index. So for starch, normally we use three different terms. One is rapidly digestible starch. Rapidly means it's very quick releasing starch. The starch is releasing glucose in a very quick manner. Slowly digestible, that means it is slowly releasing. And the third is the resistant starch, as you can see in the red. So how we are calculating it in an in vitro kinetics, whatever glucose is released in the initial 30 minutes, as you can see in the red line here, so that is considered as the rapidly digestible starch. Okay, so that could be a denatured form of starch, that could be the amorphous loosely form of starch, it could be anything. But those starch is releasing very quick, fast glucose release. Then you have a bigger window, 40 to 240 minutes, where the release is happening in a very slow manner. So that we call it as slowly digestible starch. Okay, so the first 30 minutes cause your glycemic index, and the 40 to 240 cause your extended glycemic index. Okay, so we should consume. So we all are consumers and we all have a diet of our own. So for being healthy, we have to eat food which have an extended glycemic index, like millets. Okay, because we are not having a sharp glucose spike there in the 30 minutes. Your glucose release is happening in a very slow and controlled manner, which is very important for diabetic people and also for preventing diabetes, because whenever there is a sharp peak, it is causing a lot of oxidative stress there. And that is also affecting a lot of downstream, what we say that secondary complications and sequelae of diabetes. So that can always happen. So that's why we should have this kind of, which is more rich in SDS, slowly digestible, and resistant starch. So here, what is resistant starch? Resistant starch is that fraction which is not releasing glucose. Okay, it is very less. It could be like less than 4% in your diet or in your crop. But that is very good because it is not releasing glucose, but it is very useful for your gut, your large intestine, because it acts as a soluble fiber for your gut microbiome. So these days there is a lot of outcry about the importance of gut. So I am not going to detail that how gut is important for you. From COVID to, I think, most of the lifestyle disorders, we know that gut is the second heart or second brain, and gut has a lot of importance. So the health of the gut is well correlated to all these metabolic syndromes. So if you consume resistant starch rich, that is soluble fiber rich foods like millets and some particular pigmented rice, which we have seen. So this soluble fiber content will help the growth of good microbes in your gut, and as well as it release those kind of short chain fatty acids, which is very very good for your different kind of attributes, the colon health. It avoid gut dysbiosis. There are many issues which is normally happens because of our diet that could be avoided by this kind of a diet. So that's why starch. So when you see starch, now you see starch as a RDS, SDS and RS, and try to consume more an SDS and RS. And here the scope of research is, can we find cereals which is rich in SDS and RS? Then it is definitely going to be better for diabetic people. So that what I mentioned, that we need this kind of a curve in our food. So if such kind of a staples are not available, so can we make such composition? Okay, so that also we are trying in our next CRP platform project, which Dr. Ranjan is heading. So in which we are trying to make a mix or composition of flours in which we can alter or tweak this kind of a glycemic response of our food. So these are the different terms. I hope you all are very aware. What is a glycemic index? What is a glycemic load? What is a glycemic potential? And what is a glycemic response? So glycemic index is a ratio. Glycemic is load is when you always also take that how much quantity of food you are taking. Okay, whether you're eating 100 gram or whether you're taking a 500 grams. So then you calculate the glycemic load. But when you discuss about a crop, we use the term glycemic potential of the crop. And ultimately, you have to see the glycemic response, because the response that happens in my body and your body is entirely different. Because there are a lot of extrinsic parameters also involved from the start, that how you chew your food, how your enzymes are metabolically active, and genetic parameters. So there are many things. So glycemic response is the ultimate, but there are a lot of limitations because of the ethical concerns. The studies are limited. So hence we need a high throughput in vitro method, which can, in a way translate to that end. And that's why we are doing the starch kinetics today, in which we can just assess that whether we can tell that this particular commodity is low, medium, or high glycemic. Okay, that we can do with the in vitro method. So, and recently the publication of American Journal of Clinical Nutrition also highlighted the fact that along with the starch quantity, the quality means how much RDS, SDS, and RS is present. That is also very important in your diet. So that's why you can see lot of functional foods these days are coming with high RS type or gut friendly or diabetic friendly, with having enriched fibers and so on. It's all because because our diet needs, because of the dietary transition that happened over the time and the lifestyle changes that happened over the time. So now we have to take supplements, or we have to take these kind of food that is rich in starch, quantity, less in quantity, and better in quality. So this is just a simple, the illustration which explains again that how RDS, SDS, and RS is in our body. So the initial spikes is your RDS, and the last fraction that I said, that which is very good for your gut microbiome, is your resistant starch. But the main fraction where you can tailor, where you can manipulate the glucose release, is your slowly digestible starch. So internationally, food engineers are now trying to make new food prototypes which has higher SDS fraction and low RDS fraction. So a lot of possibilities for food engineers and food technologists in this domain. So as a biochemist, so how is my role here? So what I can do is to reduce the glycemic response. So for that, I have to understand that what are the factors that convert starch to glycemic response. So we got to know from the literature that when we consider a cereal or a crop, because we are working in a plant research-based organization, here we have to understand the crop first. Okay, then only the subject or a person. So here I want to say that there are a lot of intrinsic parameters in a crop which determines its glycemic potential. And along with that, there are a lot of extrinsic parameters, as I already said. Your chewing, your metabolic rate, how active you are. Some people can be a sedentary person, some can be very active. So the metabolic rate is different. Your gastric emptying, how fast your food is moving from one chamber to another. It is also an individual centric parameter. Then what is your pre-meal? Means what is the gap between your meals? Okay, what is the gap between your first and your third meal? So all these studies have found that all these parameters and gene expression rate What is the copy number of salivary amylase in your body? What is in my body? So it's different. So we cannot work on these extrinsic parameters because it is individual centric unless you have a nutrigenomics platform, which is not yet established in our country. So what we can do is more into the intrinsic parameters to understand our crops and how the different parameters in the crop is causing the glycemic response. For example, how the microstructure. So microstructure means for every crop, we know that if you see a grain, if you take a translation of a grain, we know there are different outer layers, the pericarp, the epidermal cells. So how thick is it? So that the, the enzymes cannot penetrate. So what is the microstructure of your grain? What is the molecular structure of your starch? What is the chemical state of the matrix? How much starch is present? If one commodity have 90% and another has 50%, definitely this will be more glycemic. So what is the composition? What is the type of the matrix? Like it is only starch, or it is a predominant starch, or the starch is embedded with other components, and what are the interactions among them. So all these we can study, and with that hope we started this food matrix studies in pearl millet, which we already doing in rice, but rice was more a homogeneous matrix for us. So we introduced more studies in pearl millet because pearl millet was very good with a lot of complexity in the matrix. And then by understanding the pearl millet, we took consideration of these parameters to understand in detail so that we can say that with conviction that pearl millet is diabetic friendly. Okay, so we planned certain experiments to understand the microstructure, the ultrastructure. The ultrastructure is also very important. We should understand that how big is a starch granule. Bigger the granule, easier the digestibility. Okay, and many more. They have also observed some starch granules also have a pores on their surface. So if pores are there, these enzymes, your amylases, can penetrate very easily inside. So if pores are there, definitely it is more digestible. So understanding the ultrastructure of the granule is also very important. What is the content? What is the composition? What is the starch structure in terms of its crystallinity index by XRD? And then what is the other metric components like lipid and fiber that is present. So this was the experimental plan that we performed under this program. And we observed that even though it is polygonal starch granules, some of it has pores on its surface, some landraces, which we found later that they are sweeter because of their easily digestible nature. But most of the pearl millet didn't have any pores on its surface, and mostly they were in a complex granules. So when we have a complex granule, it is less digestible. So that could be one reason that the pearl millet is releasing less glycemic response. And other than that, we also understood the crystallinity, where we found certain pearl millet varieties, when we screened, also has a combination of crystallinity. That is, it has A and B type. So that could be also one reason for its low digestible nature. So let's now discuss about each of the parameters in detail. So as I mentioned, we always compared this with the most common glycemic commodity, which is notorious for diabetes, that is rice, white rice. So we compared pearl millet with rice as a model. And as you can see, this is the microstructure. So what we do here is we fix the grain in formalin, and then we make the paraffin blocks, and then we do section, and then we do staining. And by staining, we will get to know that what is the anatomical, the structure of. So from here, we can understand that how the pericarp thickness is there, and we can also understand what is the size of the endodermal cells and what is the size of the granules. So all these details we can get to know by microstructure study. Then we also designed this kind of, how the food metric, the microstructure is different. One of my students did work in which she said that how it is different in case of pearl millet. So we can say that pearl millet, the pericarp thickness is more, endodermal cells size is less, and also, so when it is less, that means enzyme cannot absorb on its surface more. Okay, but in case of rice, we see the pericarp is thin so that it can very easily penetrate, and the granule surface is also more so that enzymes can bind to it more. So that way digestion will be also fast. And the second we did is ultrastructure, where we performed with scanning electron microscopy, because it is known that enzymes penetrate inside and digestion happens from inside. There is an inside out digestion that is happening in our body. So for that, we did certain SEM analysis, as I mentioned. Then We also did this experiment in rice where we cross-section to see the complexity, where the amylose and amylopectin packaging of the starch granule, what we saw, it also vary according to the amylose content. So higher the amylose, there is a correlation established in some papers that when amylose is more, resistant starch is more. But it is not always true. But in certain cases, we have seen, yes, it is true, because when amylose is more, compact packing is more, as you can see here. This is white rice, this is black rice, and this is red rice. So red rice, we studied Nyavara, which is a GI tagged rice from Kerala. So this is very good for diabetic people. So we also wanted to know why it is good for diabetic people. So if we see the cross section, we can see that this kind of a structure, because here the amylose and amylopectin packaging is so coarse, so compact, so that is, and you can see the granules also in this image, in the eye session, that they are so compact, so the digestibility is very less. Okay, so that is what we got from our ultrastructure experiments. And as I already mentioned, that the three questions in our belly when we started the pearl millet program is why pearl millet is diabetic friendly. Second is why some of the land races, for example, Jafrabadi Bajra, has been known to be sweeter. Okay, so the next question addressed to me that why it is sweeter. Can you understand the basic? Because biochemists always address the question why. So the next question is why is it more sweeter? So the third is along with the starch, whether the phenolics in Bajra also has any anti-diabetic potential. Okay, so I already mentioned about the food structure. So we again compared, as you can see the thickness here in compared to the pericarp, the pearl millet is thicker. And as I mentioned, the pericarp thickness, endodermal cell bodies, and the granule size, you can see, we analyzed and we found that it is pericarp is thicker in pearl millet. That's why the enzyme cannot penetrate inside. In composition also, rice, it is very high, 80 to 90%, but in pearl millet, it is 60 to 70% starch. The soluble sugar content is almost, we can say, in the range. Amylose content is also, there is no much variation in case of pearl millet, what we got. It was like within the range of 25 to 28. But in rice, we had a lot of, spectrum of amylose containing varieties. And resistant starch definitely in pearl millet was found superior compared to rice. So this is another experiment which we mentioned about food metric interaction by confocal microscopy. So confocal microscopy, we know that by laser scanning, we can understand the metrics, and there by using the specific dyes, we can tag your macronutrient. For example, by FITC, you can tag your starch. By Nile red, you can tag your lipid. So here what we did is we tagged and then we did the confocal analysis. And in confocal analysis, you can see these red drops. These red drops are the lipids. And these lipids are present high in pearl millet. So when lipids are more, the lipid and starch can bind together and it can form another type of resistant starch, which we call resistant starch type 5. Okay, so that could be another possible reason in pearl millet, which is causing a low digestibility. And this is the XRD experiments, which I mentioned, where we perform XRD, we get the crystallinity peaks, and from the peaks, we can calculate the crystallinity index. So in pearl millet, the compact crystallinity is more compared to rice. In rice, it is more amorphous crystallinity. Amorphous means very loose. If it is loose, it can digest very fast. So that is what next scientific hint that we got towards pearl millet's low digestible nature. Then today we are doing the kinetics, which is a quantitative method, but we also want to know qualitatively how the matrix is getting loosened. To see that, we did an iodine-based analysis. So here you can see in the first 0 minute pre-digested, the matrix was so complex. It was a meshy, it was a matrix. And then during digestion, it gets loosened up. And after post-digestion, you can see it gets so dispersed. Still, you can see some because only starch is digesting. The rest, your protein or your fiber will be definitely there. So you can see in the after 240 minutes, here again the matrix is loose, completely loose. But there is a difference in rice and pearl millet, and that correlated very well with our quantitative starch hydrolyzation kinetics, which we are going to do in the practicals today. So to conclude that from these experiments, what we got to know that the complexity of the food matrix structure the interaction, the molecular configuration. That is the main attribute that is limiting in pearl millet compared to rice. So all those traditionally known cereals or traditionally known crops, which is known to be low glycemic, we have to understand. Then we have to scientifically validate in a systematic manner like this, so that we can even also elevate the status of the grain as diabetic friendly, and then we can also promote its export potential. So scientific study, that's why it is very important, and hence under this project we did some work in this direction. So the second question, as I mentioned, was why pearl millet land races are sweeter. And I think our Honorable Prime Minister also mentioned about the Jafrabadi Bajra, and then it also got a lot of limelights that this Bajra is sweeter. So as biochemist, then the question was on our shoulders that we have to know that why it is sweeter. And sweetness comes because of two components, as you all know. It could be the reducing sugars that is present, or it could be the easily digestible starch, because the starch is so easily digestible, it is digesting in our mouth, and we have started feeling sweetness. So on that direction we started, and we found that the reducing sugar percentage in among the land races we screened, the Jafrabadi had a superior, around 2.75% sugar percentage content. And among the sugars, I want to mention that glucose, sucrose, and raffinose and stachyose is also present in pearl millet. And other than that, you see the total starch content is also high, so that the starch can be degraded and easily digestible. And the amylose and amylo content is also mentioned here. So I think the balance that is happening between the glucose and the starch. So it might be happening something at the assimilatory level in the plant itself. So in the plant, we have to study the genetics of the plant to understand more why more sugar is present in the matrix compared to the storage form of starch. So that studies have to be done in future. But here we can mention that the total sugar content might be one possible reason, along with easily digestible starch. So for the starch, again, we performed certain studies like XRD studies, in which we found the crystallinity type of Jafrabadi Bajra. The crystallinity was found to be A type. As you can see, the first here, this is a orthorhombic structure, which is a very loose structure so that your enzymes can digest it fast. And in this comparison, you can see in the B image. So this is the A type crystal XRD gram that we see. And here you can see the comparison when we compared with Dhanshakti, Pusa, Chadi Bajri, and Chanana Bajri, Damodara Bajri. There we found that the Jafrabadi was having the least crystallinity. So the least crystallinity means the maximum amorphous starch. So maximum amorphous starch means easily digestible starch. So here you can see the crystallinity degree. So Jafrabadi has the least crystallinity degree, and hence we can say that Jafrabadi has easily digestible starch compared to other pearl millet varieties. So finally, we did the kinetics where we simulate the oral, gastric, and the intestinal digestion, and we did 70-75% compared to other pearl millet genotypes. Other pearl millet genotypes ranges from 45 to 60, but this landrace is having a superior digestibility. And here another term is inherent glycemic potential. So inherent glycemic potential and resistant starch both plotted in the same graph. So resistant starch is ranged only till 3%. But inherent glycemic potential, you can see here, it starts from 50, but in Jafrabadi, you can say that it is blue, you can see around 70, 75. So the blue is the glycemic potential and red is the resistant starch. So resistant starch is also around 1.5%. It is also less. Okay. So the resistant starch is less. So that means sugar response wala starch portion is less and glycemic potential is more. So this was the explanation that we gave from our side, that the high glycemic potential, the low resistance starch, and the higher sugar content could be one possible reason for Jafrabadi, which is found to be sweeter. And we finally framed this kind of a figure in which we can mention that the presence of the starch components and as well as the sugar and its starch forms like the crystalline form. So the low crystallinity lead to the sweet perception. which contribute to the through the sugar and as well as from the starch. So the sweetness can also come from the sugars and also from the starch. So here you can see free sugars come from the matrix, and also sugar, the black, that is glucose, is coming from the starch through here. Okay, so this is a digestible starch is giving sugar, and here it is different type of sugars that is also we taste when we sense the taste of the landrace, that is Jafrabadi bajra. And after the digestion, definitely the glycemic potential was more and the resistant starch was less. So to conclude this, the model shares the insights that it's one example that we can understand that how this particular landrace can be sweeter. This study can be extrapolated into different dimensions. So this we communicated to International Journal of Biological Macromolecules. So here we address two questions: why pearl millet is diabetic friendly? The second question is why it is sweeter, some of the land races. So the third question, which is also revolving around in the same domain, is the pearl millet is rich in phenolics, whether the phenolics also have any role in anti-diabetic potential. So recently we published a paper in food chemistry where we underlined the different role of phenolics in pigmented rice towards diabetics. So in the same direction, we also thought to understand what are the phenolics that is present in pearl millet. So in pearl millet, there are different type of phenolics. There are simple phenolics, complex phenolics. There are phenolic acids. There are flavonoids. So there is a different phenolic pool that is present in pearl millet. So by extracting these phenolics, so here because it is a cell line based studies and so on, here we cannot do as a matrix itself. So here we have to extract the phenolics, then we have to perform the analysis. So what we did is from this image, which we published in food chemistry, I want to mention that the glucose that is present in our blood is contributed mainly due to the starch. Here one term nutraceutical starch is coming because nutraceutical starch is the term. Dr. Sunil, can you please mute yourself? Thank you. So nutraceutical starch is a combination of slowly digestible starch and resistant starch, which I already said, it is very good. So as just telling that SDS and RS, we can in toto, we can say it is nutraceutical starch. Okay. So the nutraceutical starch, if it is very high, it can definitely cause less glycemic response. So all these hexagons, these are the glucose. So the glucose is less. But whatever glucose is released in our body, it has to be uptake into the cells by the GLUT transporters. So these GLUT transporters are low affinity transporters. So these GLUT transporters, if it is uptake inside the cell, then there is a homeostasis of glucose that is happening. So there is a glycogen synthesis happening, gluconeogenesis happening. So it's all based on the trigger glucose molecule. So all these we can study, then only we can confirm that the phenolics have a role in anti-diabetic potential. So in that direction, we performed certain inhibition assays first, because first we have to see while eating whether the phenolics in the pearl millet is inhibiting your amylase or not. Okay, and we found that definitely these phenolics have an inhibition potential because they have a possibility of irreversible binding to the catalytic nodes, and that's why these enzymes are no more active. So when the enzymes are not active, they cannot digest the starch in that way. So that is happening in one direction. The second is, excuse me for 1 second. Yeah, so second is inhibition assay, and for that we also help support of certain docking analysis, which we want to do because we want to know in the extract what are the phenolics actually that is inhibiting. So for that, already some of the molecules were known in pearl millet, so we took them and then we docked it, and then with docking they understood that. Just a minute. ठीक है नहीं okay. Okay, sorry for that. So To continue, so what we did is with IIT is that we did certain docking in which the phenolics we tried to dock and see that whether what are the possible phenolics present in pearl millet is inhibiting this kind of an enzyme. So then we found certain like luteolinic acid, malatoic acid, and salicylic acid. This could be possible. So first, after identifying this, we can use this as biochemical markers for screening in future. The second is similar we did with the glucosidase also, because it is present in our intestine, which converts the maltose to final glucose. So there also we found that the phenolics have a role in inhibiting these amyloglucosidases. And the same strategy we performed. We docked the glucosidase structure that is available with the different phenolics that is present in pearl millet. And we found there are more than around 10 to 12 phenolics like flavonoids and phenolic acids, which were inhibiting. And these are all phenolics that is confirmed in pearl millet. Okay, we only took those phenolics that is already reported in pearl millet matrix so that we can confirm that whether they are really doing the inhibition role. And this is another very interesting study we did. This is called radio tracer assay, in which what we did is we did use of some hepatocyte cell lines, which is a liver cell line. And because liver, we know that is a real hub of your glucose homeostasis that is happening, which convert into glycogen and all other things that is happening. So we took the liver cells and we used radioactive glucose also. So the radioactive glucose, what we did is like in presence of radioactive glucose and the phenolics, we analyzed how much glucose uptake is happening. So that's how we know that how much phenolics is modulating the transporter. In presence of phenolics, it is taking less glucose inside. In sometimes without the phenolics, it is taking more glucose inside. So based on that, we can also go to an understanding that the pearl millet phenolics is not only inhibiting the carbolytic enzymes, it is also having a role in regulating the GLUT transporter modulation. So then again, we checked what are the phenolics that is modulating the GLUT. So by docking, we got to know some list of phenolics that is doing GLUT2 modulation. And finally, we did a lot of docking to understand whatever phenolics that is reported till date in pearl millet, to understand that what is having an inhibition role and as well as modulation role, so that we can pinpoint some of the phenolics present in pearl millet has an anti-diabetic potential. But these are just prediction. So the next step is how to confirm. So for confirm, we need NMR support. So we have a collaborator in MG University, Kerala, with whom we performed certain NMR studies, and we confirmed the presence of 2 major flavonoids and phenolic acid in pearl millet, which is having anti-diabetic potential. So to conclude the whole study, we can say that the phenolic extract with enzyme guided assay, along with the in silico studies, identified 2 different flavonoids like luteolin and acacetin that is present in pearl millet, along with phenolics like malillotic acid and salicylic acid, because based on the docking score and as well as their presence in NMR studies, confirmed that the presence of these 4 in pearl millet has an anti-diabetic potential. So first, we understand that how starch is contributing towards the diabetic friendliness. Now, the millet phenolics also have a role. So that means it's a boon from both sides. So that's why we should have millet this thing. And whatever we studied with our curiosity, Shelly ma'am already mentioned this in inaugural session, that one of my student, this is a confocal image. So we modified this image it into a science art, and we are indebted to INSA INEAS because they announced this award on the National Science Day, and we are grateful for that. And along with that, we also because science is always a fun and it's an inner calling, that's why we are doing. So we also do a lot of science cartoons, SciToons, and this kind of science reporter articles, because many of you are extension scientists. Whatever our research we do, it should not be within the 4 walls, and it has to reach to the stakeholders. And consumers are one of the main stakeholders, even though it's a farmer or an industrialist, all are consumers. So because this topic has a lot of importance in our daily life, so we write a lot of articles in common journals like Science Reporters and so on. And through INEAS, which is a wonderful platform for science outreach, we go and meet students in rural and other areas, and we also make them aware about what is, you have to be abreast with the nutritional research of this decade. That's how we look out to the wonder of pearl millet metrics. And these are some more papers, because we have a very strong rice starch quality team also in our division. And if you want to get more details, you can go through these papers. And last but not least, I think I'm a bit on a higher side on my time, but I hope that whatever we did, and whatever we are more planning in this direction, I think that will add more novel insights into the pearl millet and the crops which are not much unraveled till date. So that's our humble try in this direction. And last but not least, I would like to thank the Pearl Millet Group and Head Biochemistry, Nish team, and ECRIP Jodhpur, Dr. Tara Satyavati and her team, and also some other collaborators for their immense support. And this is my rice team, a bit more diverse, because we have been working for more than 5 years now. And I will be now proceeding for my work in Purdue also in this direction. And that's me in a nutshell. And I think now anybody has any questions, it's always welcome. Thank you.
Ma'am, the glycogen and the starch, when we estimate by hydrolysis and we calculate the glucose content. What kind of factor we can use to convert the glycogen availability in the meat and the starch availability in the grain?
So glycogen I have not done, and I am not aware because we never did it. But starch normally, see, if you are already only mentioning about the glycemic potential aspect, then you have no need of any conversion to convert it into starch form. So normally the fraction of 0.9 multiplication what happens when you want to convert the glucose release into starch conversion. But normally, ideally, it should be like how much glucose is released after the time point by total available carbohydrate. Because in the metrics, sugar is also there, starch is also there. So it's a total available carbohydrate. So you have to digest the whole food metrics, whatever you are taking with Anthron method or something, estimate it, the total glucose. Then you have to calculate from your this thing, and then you have to take a ratio of it. So that is what you have to do. Because when you do an Anthron method, it is total digestion. But when you do an enzyme-based method, it is a human mimicking digestion. So definitely it will be less. So on that basis, we have to calculate.
Okay, initially we have to extract the starch and keep it aside, and then
No. You have to take the food commodity as a whole. That's why I mentioned the importance of food metrics. If you are taking a cooked rice, take the cooked rice, mash it, and use it as a food source, just like you are eating. The mashing is the chewing simulation. So you have to take it. Pearl millet roti, you have to mash the roti itself. So 30 seconds is considered as the average time of our chewing. So normally we do that much, say similar kind of chewing or mixing. Then you have to do for chamber-wise digestion, amylase, pepsin, and then your glucosidase pancreatin. So that, and after that 240 minutes, you can take and you can see how much glucose is released. So that again, actually, ideally you have to again do the diffusion through a dialysis membrane, which sometimes we are not doing because glucose is definitely going to be bioavailable considering that. So that is the bioavailable glucose. If you isolate starch, definitely it will be more.
Okay. All right, ma'am.
Thank you.
Hello. Hello.
More question?
Hello. Am I audible?
Yes, sir. Yes, sir. Please.
Dr. Veda, it was a nice lecture. I just would like to ask you one thing, that we have biofortified bajra, like iron and zinc rich bajra. Isn't it?
Okay.
Hello.
Yes, sir. You're audible, please.
Yeah. So my question is, what are the different phenolics or flavonoids which are responsible for making bioavailability of this iron and zinc? Because even though you increase the iron and zinc content, bioavailability is a major concern.
Exactly.
soybean, some flavonoids have been identified. Do you have any information? What extent they are made bioavailable?
Yeah. May I know who is this asking? I cannot see your name. Okay, sir. Sir, till now I have not seen any this kind of a study which can clearly say that because of this is happening. But what I understand is the minerals are also present in the matrix. So if phenolics are more there, so it is going to, it is like an entrapment kind of a thing that is happening. And the first, because of the entrapment, most of the nutrients are not able to release into the matrix, from the matrix. So if it is not released, then it cannot be bioavailable. So the bioaccessibility is the major problem. So when phenolics is there, mostly it is not phenol, it's a polyphenol. So polyphenols are all big structures. So they always have known as, they form caging. So they cage these minerals and other small molecules inside their cages. And because of this phenolic presence, these minerals are not getting bioavailable in our body, because most of the polyphenols are also not digestible in our body. So these things actually happens. So that's why I say that when we estimate only minerals, it is different case. But if we estimate the bioavailability of the minerals, then, and many more, there might be the presence of phytic acid, which is again an absorption inhibitor in the matrix of pearl millet. So again, it is going to reduce the bioavailability. Phytic acid is negative cloud, so it is going to chelate the positive minerals. So there are different reasons in matrix which can affect the bioavailability.
No, my question was, because we are making efforts to increase or improve the through biofortification program. And on one side, you said that phenolics have antidiabetic role. And at the same time, they are not making the minerals available. So it is a sort of thing. So exactly what is the factor which is responsible?
Sir, if your objective is both, then we have to find that what is the balance of it. My objective was not to release the minerals. My objective was to understand the bio availability towards this thing. No, I got your point, because we are enriching in a biofortification mode. So the plant is increasing the content of the minerals naturally in the matrix, even if you're increasing 10 to 100.
Yeah.
But, and in every body, the rate of absorption and everything is also different, sir. Even if you do increase to 100 ppm, it is not necessary that it will be absorbed in that rate. So it is not that easy or that simple to say that we increased or biofortified to this level, so it bioavailability also. So that's why all these biofortification studies has to end with a bioavailability. Until unless there is no point in mentioning the quantum jump in the numbers or the folds.
Thank you.
Okay, then if there is no more question, is it?
Ma'am, one more question I have. Can I ask one?
Yeah, please.
Ma'am, you said no, the initial stage of digestion will lead to oxidative stress in the gut. Can you
rapid spike of glucose? Yeah, the rapid spike of glucose, it is known to have this thing, because whenever there is a rapid spike, so this is the problem of insulin sensitivity happened, people have observed because of this rapid spike. So that's why we say high glycemic commodity whenever you eat the junk foods. So there is a rapid spike of glucose in our body. Okay. So then the body has to have a mitigate mechanism. It has to release more insulin. So this kind of a imbalance that is happening more and more. So that has known to have a lot of oxidative stress in liver. So there are reports on that.
So junk food is other side, ma'am. Junk food.
No, junk food means carbaholic food. What I mean.
Fried.
No, carbaholic junk food. Your soft drinks or whatever you are taking, no, which is rich or easily refined foods which release your sugar in a very fast manner. So all these things has known to have this kind of oxidative stress, and people have seen the beta cells have destroyed because of this stress.
Okay. Thank you, ma'am.
Hello.
Yes, Dr. Sanjay.
Madam, very nice lecture you have given. Can you please suggest one or two varieties for central zone, biofortified varieties of pearl millet?
I think varieties, I think breeders are there. They will be best to suggest. I am a biochemist.
I'm asking for all the groups in house. Please suggest some because nowadays it is very important to promote the biofortified varieties.
Okay. I think Dr. Tara's lecture is going to be Dr. Sanjay. Please shoot this question there. She is the best person to help you.
Thank you, ma'am. Thank you, ma'am.
Okay. Yeah.
I think we all agree that it was an excellent talk by Veda. She's very gradually built up the whole story of importance of starch and the availability of starch. Excellent, Veda. It was very good.
Thank you so much, ma'am. Thank you, ma'am.
So over to Suneha now.
Dr. Archana Sanjay, good morning.
Good morning, sir. How are you? So nice to see you.
Okay, so you all agree that, and ma'am also mentioned that it's a very good talk, very informative, and Dr. Veda explained very nicely about the starch matrix and how it is responsible for means for some of the genotypes that they are bitter in taste and some are not. So what is the reason behind that? So very nicely she explained about that. So thank you, Dr. Veda, for such a wonderful lecture. So now the next session is the practical session, and that is also by Dr. Veda. So
do we have a gap or it doesn't continue?
There is a gap.
Okay, thank you.
15 minutes at least.
Okay, that will be great.
If it's agreeable to all the participants.
Yeah, definitely. So we can meet after 5 minutes.
Half an hour?
Half an hour, it's okay. Yeah. So after half an hour again.
By 12 we will start. 12 we will start.
Okay.
Yeah, that would be
Thank
great.
you so much.
12
Thank
is
you.
okay.
Thank you, madam. Thank you.
Thank you. It was a good talk, ma'am.
Thank you so much.